Most people assume the immune system simply becomes weaker with age. T cells become less responsive, infections become more dangerous, vaccines can work less effectively, and cancer risk rises. But people who survive beyond 110 years old appear to break part of that rule. A new Cell Report study published on August 19, 2026, found that supercentenarians do not merely preserve a younger immune system. Instead, their immune system appears to reorganize itself. A normally rare population of immune cells called cytotoxic CD4 T cells begins expanding dramatically around extreme old age, sometimes until one single clone dominates more than half of that entire cell population. That finding changes the question scientists are asking about healthy aging. Perhaps the secret is not preventing the immune system from changing. Maybe successful aging sometimes requires the immune system to change in exactly the right direction. CD4 T cells are normally described as helper cells. Their traditional role is to coordinate immune responses by communicating with other immune cells. CD8 T cells are usually the killers. They recognize infected or abnormal cells and destroy them using molecules such as granzymes and perforin. Cytotoxic CD4 T cells are different. They still carry the CD4 identity, but they acquire much of the destructive machinery normally associated with CD8 cells. Earlier work from the same research group showed that these unusual cells are exceptionally abundant in people over 110. In that study, supercentenarians had dramatically more cytotoxic CD4 T cells than younger controls, and the cells expressed genes involved in killing abnormal targets. The new study asked what happens before and during this expansion. Researchers used single-cell immune profiling, combining gene expression measurements, surface proteins, and T-cell receptor sequencing. Instead of looking at T cells as one population, they could follow individual cells and determine both their functional state and their clonal ancestry. The resulting data suggests that cytotoxic CD4 T cells begin expanding around the age of 100 and continue to accumulate in some of the longest-lived people. This is important because every T cell carries a receptor designed to recognize particular molecular targets. When one T cell encounters something its receptor recognizes, that cell can divide repeatedly, producing a large family of genetically related cells carrying the same receptor. This is called clonal expansion. You can think of it like an emergency unit receiving evidence that one particular threat keeps returning. Instead of maintaining thousands of equally sized teams, the system repeatedly expands the team trained to recognize that threat. That appears to happen on an extraordinary scale in some extremely old people. In one centenarian analyzed in the new study, a single clone represented 53.8% of that person's cytotoxic CD4 T cells. This was not a random collection of aging immune cells. A huge fraction descended from one original T cell that had apparently received a very strong or repeated signal to expand. Normally, that kind of repeated stimulation raises another concern: exhaustion. When T cells encounter the same antigen continuously, as can happen during chronic infection or cancer, they can gradually lose function. They may express inhibitory molecules, stop proliferating effectively, and become less capable of killing targets. Aging also pushes many immune cell populations toward highly differentiated states. Yet the supercentenarian cells showed a more unusual pattern. As cytotoxic CD4 T cells matured, they sequentially lost surface molecules called CD27 and CD28, signs of advanced differentiation. But the available single-cell data suggests that this differentiation did not simply end in classical immune exhaustion. These cells remained part of a strongly expanded functional population. That distinction is crucial. Extreme longevity may therefore involve something very different from maintaining an untouched young immune system. These people have lived through more than a century of infections, inflammation, tissue injury, and accumulating abnormal cells. Their immune cells have been repeatedly challenged, yet some populations seem to adapt rather than simply collapse. The next part of the study produced an even more provocative clue. Researchers compared the T-cell receptor sequences from each person's most expanded cytotoxic CD4 clone against public databases containing T-cell receptors identified in other diseases and tissues. Nearly 36 matches were found in individuals with cancers including lung, breast, and liver cancer. None of the centenarians or supercentenarians in the study had been diagnosed with those cancers. That does not mean the researchers discovered hidden cancer inside these people. A matching or similar T-cell receptor sequence does not automatically prove two T-cells recognize exactly the same tumor antigen. T-cell receptor specificity is complicated, and different targets can sometimes generate related receptor patterns, but the overlap raises an intriguing possibility. Some of the massively expanded cytotoxic CD4 cells in extremely old people may be responding to molecular targets also encountered inside tumors. Lung cancer was particularly interesting. The Associated Genomic Archive reports that expanded CD4 cytotoxic T-cell clones shared CDR3 beta sequences with tumor-infiltrating T-cells, especially those identified in lung cancer. CDR3 is the highly variable region of the T-cell receptor that plays a major role in recognizing antigens. In simple terms, researchers found similarities between some of the immune receptors dominating the blood of exceptionally old people and receptors previously found inside tumors. Why would this matter more after age 100? Because the biological environment changes with age. Mutations accumulate. Senescent cells become more common. Cells experience decades of DNA damage, viral exposure, metabolic stress, and inflammation. Even people without clinically diagnosed cancer are likely producing abnormal cells throughout life. Most never become dangerous because immune surveillance and other protective mechanisms eliminate them or prevent expansion. The researchers propose that cytotoxic CD4 T cells could represent one adaptation to this increasingly abnormal cellular environment. As first author Kosuke Hashimoto explained, aging creates more senescent and potentially cancerous cells, and immune adaptation to those changes may contribute to exceptional longevity. There is some biological support for that possibility. Cytotoxic CD4 T cells can contain granzyme B and perforin, molecular weapons capable of damaging target cell membranes and triggering cell death. Earlier experiments in supercentenarians also showed that these cells could produce inflammatory immune signals including interferon gamma and TNF-alpha after stimulation. Other research suggests these cells may also respond to persistent viral infections. Cytomegalovirus, or CMV, is particularly important because it can remain in the human body for life and strongly reshape the T-cell compartment. Recent research in human lungs found that CMV preferentially drives accumulation of cytotoxic CD4 T cells, supporting the idea that repeated antigen exposure can push helper T cells toward a killer phenotype that creates two possibilities that may even overlap. These cells could be adapting to chronic infections. They could be responding to abnormal host cells, such as cancerous or senescent cells. Or the same long-lived immune population could help with both jobs. But there is a major complication. Cytotoxic CD4 T cells are not always beneficial. They also increase in some autoimmune and inflammatory conditions. Highly differentiated T cells can contribute to tissue damage, and immune responses that are useful against infections or tumors can become harmful when directed against normal tissues. Reviews of these cells emphasize that their effects depend heavily on context. The same cellular machinery that could improve cancer surveillance might also contribute to inflammation or autoimmunity under different conditions. So why do supercentenarians appear to tolerate such enormous expansions? That may be the deeper mystery. Longevity might depend not simply on producing more powerful immune cells, but on maintaining enough control that those cells attack the right targets without causing destructive chronic inflammation. This idea fits a broader shift in aging research. Immunosenescence is often presented as straightforward deterioration, but immune aging is increasingly understood as remodeling. Some populations shrink, others expand. Naive T cells become less common while experienced clones accumulate. Certain inflammatory pathways rise, yet some exceptionally old individuals retain surprisingly functional immune responses. A 2026 study of 160 Colombian centenarians reached a similar conclusion from a broader perspective. Researchers found substantial variation in immune aging between centenarians and argued that their data challenge an overly simple model in which extreme old age necessarily equals universal immunological collapse. The new supercentenarian study goes further by showing what successful remodeling may look like at the level of individual T-cell clones. But it does not prove that cytotoxic CD4 T cells cause these people to reach 110. That limitation is essential. The researchers studied people who had already achieved extreme longevity. This creates a classic problem in longevity science. If a biological feature is found in exceptionally old individuals, it could have helped them survive, or it could simply be something that develops after surviving that long. The direction of causation is unclear. Perhaps these unusual immune cells protected them from cancer and infections for decades. But it is also possible that surviving to 100 created the repeated antigen exposures that caused these cells to expand. The study also examined circulating blood cells. We still do not know whether the same clones behave identically inside the lungs, liver, skin, brain, or other tissues where cancer and senescent cells actually emerge. The researchers have identified tissue behavior as one of the important next questions, and although some T-cell receptors resembled those found in cancer datasets, the precise molecular targets of the supercentenarian cells remain unknown. Scientists would need to determine which antigens each clone recognizes and then experimentally show that those cells can kill the relevant cancerous or senescent targets. Until then, it would be inaccurate to call these cells proven anti-cancer cells responsible for longevity. There is also no supplement, diet, exercise routine, or drug currently known to reproduce this specific immune configuration in healthy humans. Trying to indiscriminately increase cytotoxic T cells could be dangerous because immune activation can produce autoimmune disease, inflammatory damage, or other unintended effects. The practical importance of the study is therefore not a longevity treatment. It is a new model of what successful immune aging might mean. For years, the goal sounded simple: keep the immune system young. But a 110-year-old cannot literally have an immune system that has experienced only 30 years of life. It has accumulated more than a century of immune memories and repeated challenges. The remarkable feature may instead be adaptability. These supercentenarians appear to have taken one normally uncommon class of helper T cell and expanded selected clones into a large cytotoxic force. Some of those clones resemble immune cells observed in tumors. And despite their extreme differentiation, the population does not simply look like an exhausted immune system that has stopped responding. That gives us a very different picture of longevity. Living to a 110 may not require freezing the body in a youthful state. It may require continuously rebuilding biological defenses as the threats change. At 20, the immune system needs flexibility. At 60, it needs memory. And beyond 100, perhaps one key advantage is the ability to take the few immune cells that still recognize the most important threats and expand them aggressively enough to keep those threats under control. Scientists still need to prove whether that is what these cytotoxic CD4 T cells are actually doing. But if they are, extreme longevity may depend not on avoiding immune aging, but on mastering it.